Okay, welcome to SID3020 Natural Products and Biotechnological Processes. So I'll be covering, I'm sure Prof. Khalija have already mentioned it to you, but I'll be covering the topic on biotechnological processes. So what I've prepared is, or what I'm doing is actually to divide this, all my lectures into three main topics. So the first topic is biotechnology. The second one is about enzymes or in vitro technology, and the third one will be about cellular, which is in vivo technology. So these are the main three topics of this subject that I'm going to try and cover. So hopefully we do have enough time. Okay, so my name is Dr. Abdullah Hadi Ahmad Fuad, for those who don't know me, but I'm sure most of you have already known me because I did see a few names or many names that is kind of like familiar. Okay, down here I have my QR code for my own website. On this website, I normally post in a few information about, well, obviously I'll post all the videos of all the lectures. I'll put it there for you guys to, go and have a look at everything, or otherwise, you can just follow the YouTube channel that I've set up for my lab. Yeah, excuse the noises, because my son is here. Okay, so this is my full name. I prefer people to call me just Hadi Fuad. Okay, if you just Google Hadi and Fuad with double A, you'll definitely see my name on top of the Google list. So why I'm teaching you this subject is because when I did my bachelor is not as what you guys are doing, Bachelor of Chemistry. So my bachelor is actually Bachelor of Biotechnology. But my major is in chemistry. So I did a lot of chemistry. I did learn a lot about chemistry, similar as you guys, first level, second level, and third level of chemistry. But at the same time, I did learn about the biotechnological aspect of our world. So my work is not just focusing on the chemistry bit, but also on the biology a little bit. So that's why I was asked to teach this subject, replacing previously Dr. Swanston. Okay, and then once I completed my bachelor degree, I did my PhD in medicinal chemistry. Also in Australia, my focus was actually in vaccine. Okay, so apologies for the handwriting. I'm using a mouse. And my work experience is a little bit about my background. I did a research assistant following my PhD. So I did work in, we call it Institute for Molecular Bioscience, again in Australia. And then went back to Malaysia in 2017, did my postdoctoral for a few months. And then I actually managed to get a post as a senior lecturer in the chemistry department. So that's when I started working in UM, which is in 2018. And then while doing my while working, I had the opportunity to actually go to Harvard and did a little bit of more training. So I have a research website. So if you just put your phone, go and QR code, and you will see this website. So I'm actually still building it. But it has a lot of information about what I'm doing, my research, my background, and also all the lectures that I'm giving. Normally, if I record it, then I'll put it there for anyone to actually see. Okay, so those are my pictures, and in Harvard, I work with Professor Joseph Brain. So he was he still works there. He is an expert in lung physiology. So I did learn a lot about cells, about in vitro technologies, in vivo technologies that we do to look at different types of what we call interaction between drugs and our own body. Okay, so a little bit about the lecture information. Throughout the slide, you will see this yellowish cartoon. It's an indicator that information is trenched. So if you actually downloaded the lecture notes and you have went through it a little bit, I have this guy at this top right corner. So any pages that has this image means that there are some information that you need to fill in. Okay, so you either fill in while you are reading the lectures, or you do it on your free time. So meaning that if you did not attend the lecture and you just download all the lecture notes and hoping that I covered all the materials there, you are definitely wrong. So my way of is normally I will trench a little bit of information, I'll took it out, and then during the lectures that I will put it in. Okay, so you only see that information if you actually listen to my lectures. So this time around, I'll add yellow guy in there on the top right corner, so that sometimes you will see a whole page is blank. There's no error. It's purposely left for me to write something. So I will try and write, especially because now I'm using a mouse, so my handwriting will be like very, very bad. So try and catch up. And if you don't understand anything, again, you can stop me anytime. I don't want to just babble alone for the next 50 or 40 to 45 minutes. So by all means, if you don't understand about something, or if you think that the information is wrong, that you have more accurate data, then stop me, and we will have a look. Okay, and then the second one is attendance link. So attendance link is available on Spectrum. So I'm not using Spectrum system for the attendance, because I had an issue where either the student were not able to log themselves in or click attend, or second reason is because once the semester ends, sometimes what Spectrum will do is to clear out all the data. So I don't want that to happen. So that's why I switched to Google Classroom, Google Form. So I did put the link on Spectrum. Okay, so use that link. You will need to key in your name and student ID. I have your time and date, so it doesn't matter. And another one is secret code. So what it means is, if you have the link but you don't have the code, then you still cannot check yourself in. So you still need to look, come, listen, and at least be present. Okay, and I'm hoping that you do not share this code with your friends. Okay, so today's code is biotechnology. So when you do your attendance link, either now or after this, make sure that you key in biotechnology as the code for this lecture today. Okay, so a little bit about lecture information. We will have 3 synchronous learning, okay, 2 lectures and 1 tutorial, and 10 asynchronous learning. So 8 lectures and 2 tests. Asynchronous means that I'll be doing a recording, and then I'll post a recording, and it's up to you guys to actually view it. I did a lot of asynchronous teaching last semester, and I did notice that a lot of students actually missed it. So this time around, instead of just doing asynchronous lectures, I'll have 2 tests in the middle. So if you miss this, then you're gonna miss your 15% from my section. Okay, so it's quite big. So make sure you keep up with all the lectures. I'll post the lectures at least on the day itself, within the time frame of the lecture itself. Okay, so I'll do that, and make sure that you don't have to watch it in some other different times. Okay, so in this 13 weeks, 13 lectures, so that is how I separate it. Okay, so today is 26, so we have one synchronous lecture, and then on the 28th, on this Wednesday, we'll have another synchronous lecture, and then following that, we'll have asynchronous lecture, meaning that I will just post a pre-recorded lectures, and it's up to you guys, and hopefully that you can watch it during the lecture hours. And then finally on week 15, week 5, okay, so I'm planning to do the first quiz here, okay, on the 5th week. So this quiz will cover the first 4 topics that we will be looking at. All right, and then mid-semester break. So make sure the mid-semester break is not between week 7 and week 8. So they switched it earlier, so it's between week 5 and week 6. So you have your mid-semester break, and then you have your continue to asynchronous lecture, another asynchronous, and then on the week 10, we will have another quiz. Okay, and this quiz will cover the 4 lectures that we will cover soon on between week 6 to week 9. So I'm not gonna ask about everything on week 10. Okay, and then we will have another 3 weeks of lectures, and then finally on week 14 we'll have your final exam. Okay, final exam. Okay, okay. So this is how the lecture or your learning session is planned for, at least for my section. So for Prof. Khalija's section, she has a different ways to do it. She has, all these information that you need to answer, all these questions that you need to answer along when you are either reading the slides or looking at the videos. But in my case, it's all videos, it's all information, and then we have a quiz, and then another information, and another quiz. Okay, and this is your final exam. We call it a summative assessment. I'll give you guys an assignment, which will be a 35% towards your final mark, and I will give it on week 13. So week 13 here, I will disperse the information on week 13, and then you will have 1 week to actually complete it, and you need to submit it on week 14. So I will give you exactly 7 days to finish it. Okay, so you should expect that this assignment is not as easy as it might sound. So it might be a bit difficult, but I haven't prepared the questions yet, so we'll see. Okay, so for today's lecture, the learning outcome is to illustrate the use of biochemical and biotechnological methods in chemistry. So with primary focus on the biotechnological section, and understand what is biotechnology all about, the importance of biotechnology, and different approaches for chemical biotechnology that we can do. Okay, biotechnology is where you can grow an ear out of a mice. Okay, so this is actually not a Photoshop picture, it's a real picture. Okay, and probably many years ago you have heard about cloning technology. Okay, so cloning is also part of biotechnology, where the name of the first clone individuals is Dolly the sheep. Okay, and finally biotechnology is where you can grow meat out of a petri dish in the lab. Okay, so this one is something that is being imagined of, so that in the future we don't have to kill any animals, or if we don't have enough animals, you can actually grow your own food in the lab. Okay, all right, so some definition about biotechnology. So biotechnology is the exploitation of biological processes for industrial or other purposes, especially the genetic manipulation of microorganisms for the production of antibiotics, hormones, etc. So we call it bioproducts. Okay, so this is the definition. If you Google it, then look for the definition under Oxford Dictionary. That's the definition. But my take of biotechnology is basically, to separate the biotechnology words itself. So bio and technology. So bio is related to using a biologics. Okay, so biological entity, enzymes, microorganism, and so on and so forth. And technology is, well, towards the end of the day, you cater for industrial purposes. Okay, so technology is normally related to in industry. So you have a computer, it's a technology. So industrial application of computers, well, we are doing it now. Okay, so using a biological form of something for a purpose, industrial purposes. In other words, using biological resources or biosynthetic processes for chemistry application. But again, this is my definition because my background is in biotechnology. For you guys, it will be catered more towards these two things. Okay, so biocatalysis and bioreactor. So that is the principle of biotechnology for you guys. Okay, so these are some example of highly sought biotechnological products. You have natural products. Okay, so for example, natural products where you can have, well, pretty much, I'm sure Prof. Alija have covered in some of her lectures about natural products. So you are well versed about natural products. And also natural products that can be converted to a biotechnological application. Currently, most important product of all, which is the polymerase chain reaction. So we all heard about PCR nowadays, PCR in doing COVID-19. Okay, so that is one of the product of biotechnology. So by analyzing, by learning about what the biological DNA replication does, so you actually try and extract out the technology and then do it in the lab. So that is a PCR machine, and that machine is actually, oops, PCR. That machine is basically doing what our cells have been doing for eons. Okay. All right. So why is biotechnology important? First page for that green, yellow guy. Okay. So there are three main criteria about the importance of biotechnology. The first one is food. Okay. In the food industry, where people use yeast. Again, this one has been going on for generations and generations, even before our modern countries were formed, to making bread, pizza, beer, alcohol, and so on and so forth. And more recently, people have been using lactase, an enzyme that is extracted from cells to produce a lactose-free milk. So people like me who are lactose intolerant, we can now drink milk. Otherwise, before this, when you drink milk, you get like a lot of uh, diarrhea and whatnot. So it's a product. It's one form of the importance of biotechnology. So for food. Second one, for material. So you have natural bioresources, for example, oil palm, rubber, where you actually do a lot of biotechnological research on that to produce more oil. If you actually go and look at, I, I don't have any plans to actually look at this in detail, but if you are interested in working with palm oil or rubber, you can actually look at or find the information about how palm oil evolved over the generation, okay? Especially in Malaysia, in our country, where palm oil and rubber are one of our most sought natural resources. So there's a lot of research going on around oil palm and rubber. Okay. Additionally, using biotechnology, you can produce a rice which is rich in vitamins. Okay. So the name of the rice is golden rice. So again, you can Google this. So what they do with golden rice is they actually included a gene to synthesize vitamin A in rice, which we don't have in our common rice, because as you know, rice is normally just carbohydrates. So researchers have actually used biotechnology approaches to modify rice to produce vitamins in the rice. So it's it seems like very tiny thing, especially for a Malaysian society, but for international, for example, in Africa, where people don't have enough vitamins, they don't have enough minerals, and so on and so forth. So this golden rice project is actually very, very beneficial for them. And the third one, under the genetic subheading, Adam, please. Okay. So you can actually, modify genetically engineered cells to produce vaccines, genetically engineer bacteria to do that. You utilize PCR for COVID-19, RNA. Okay. That one is just falls under genetic, because you are using a genetical technology. And also you can actually modify plants so that they can resist to a certain weather, like drought, for example. Okay, so these are all the importance of biotechnology. Even though you don't really say it nowadays, how are these related to chemistry? I will show it to you soon. Okay, so biotechnological diversity. So we have three main subheadings. Okay, first one is cellular technology, so which is fermentation, cell culture, a bioreactor again. So you see this bioreactor for the second time. And these are all we call as cell processing or in vivo technology. So in vivo, if you have not heard about in vivo is basically you are utilizing a cell. Okay, so you are utilizing cell. What you have been doing in the laboratory, for example, is you normally call it ex vivo or in vitro. So you're doing in a petri dish, it's ex vivo. You're doing something outside of a cell, a living cell, then you call it ex vivo. Second, diversity is genetic technology. So involve genetic manipulation, introducing new genes, as I mentioned, drought resistance, or altering or existing of processes. So we'll go in a little bit more detail when we reach here, okay, in a few weeks time. And the third one is enzymatic technology, by which we extract out all these enzymes. So we have cells that are working for a certain purpose, and normally, you know, the cell needs to have its own machinery to do that processes. And this machinery is normally the enzymes. So what we do is you extract out the enzymes, and then you can do it in the laboratory scale. Okay, so for example, biocatalytic reaction. Um, One enzyme that is, well, we'll cover it on Wednesday. So this one is just introduction. Okay, so the process flow. So how does biotechnology work? So biotechnology works is normally when you have a substrate and you want to produce, oops, you have a substrate and you want to produce a product. Okay, so this is the biotechnology stands in the middle of these two. So you have a substrate, you want to form a product. So you put it into biotechnology, and then you do a separation and you get your product. So what we are learning, what uh, we will be focusing on is these two things, which is what, when you are doing the reaction, when you are doing the process, what are the things that you need to know. Okay, so that is one thing. And second one is once you do have the process, when you have the product, now imagine that you have two different entities. How do you separate them? And because biotechnology, when you use enzymes, they are all catalysis. And as a chemist, when you talk about catalyst, it means that it's reusable. Okay, so to reuse it, how do you actually separate them? So we will cover about it. Probably Prof. Khalija will also cover a little bit because, well, this is part of her area research work. Well, I'll be giving you on the basics on the theory level, and she might, talk about based on her experience. Okay, so you have a substrate, you want to produce a product. So here, what involves is chemistry and biology, which is a biochemical processes, and normally these are utilized in a bioreactor. Okay, bioreactor is like a very huge incubator where you grow cells, and it's normally catered for industrial purposes. But for us, like me, myself, and my colleagues in we don't really use a bioreactor because bioreactor is normally for a very large scale. We use a very small scale, so we can still use enzyme to get a reaction going. Okay, so that is an example of a bioreactor. It's huge, but this one is not the biggest one. That one is about 20-liter capacity bioreactor. Okay, so this one is like a small scale bioreactor. So what you have in here is actually a cell, or not just one cell, but multiple of cells. You have thousands or millions of cells in the bioreactor, and the cell will act as the process, the factory, the source of the factory to source millions and billions of identical cells, microbes working together for one ultimate purpose. So, and the ultimate purpose is for them to produce the product that you want. Okay, so some basic information that you might need is the first one, this purple thing here is what we call as nucleus. Okay, ER is the yellow colored organelles here. We call it as an endoplasmic reticulum. Okay, well, you can always Google and find information. Endoplasmic reticulum. Okay, the cyan color is Golgi, and these purple ones around here, okay, around all the cells, this is including all the organelles, is what we call as cytoplasm. Okay, why do I want you guys to know this? Well, because as we go on, okay, when we're talking about the technology itself, there are certain bits that are important. Okay, so the nucleus has a certain function. But these four are the main system in the cell. There are a few more, like for example, this one is called as vacuole. Small ones is called as vacuole. That one is called as mitochondria. But we are not focusing on that, because towards the end of the day, this subject is for you guys to focus on what you can do with biotechnology, and these four organelles that we call it are the main ones that are very important. Okay, so again, the process flow when you have genetic technology. Okay, so it's a manipulation and editing of the nucleus. Okay, so when you want a cell to do something for you, so normally what you can do is you can manipulate or edit. Adam, please, pergi tengah ni ya. Is to manipulate the nucleus, or well, nowadays you can not, you can just ignore the nucleus and then just do the editing on the cytoplasm, but it doesn't stay long. We're not going to delve into that. So basically, genetic technology for manipulation and editing, you change the nucleus. Okay, and then second one, for fermentation, where you normally have a substrate, the cell will do all the work. So the substrate will be either ingested into the cell, change into the product form, and then excrete out. Or sometimes what you can do is you can edit it so that the cell actually gives out, so excrete out all the important proteins, so that it will form the product. So excuse my stuttering. I'm a bit lost because my son is actually playing in front of me. Okay, and the third one is enzymatic technology. Again, this one is you have a substrate, and you have the extract of a particular importance, like for example, enzymes from the cells, and then you actually do it experimentally in the lab, or a different types of bioreactor, then you can produce the product. But of course, all of these will include a separation process that I didn't show, but it's equally important so that you can get a product that has a very few side effects. Okay, so biotechnology and chemistry, where does it stand? So chemistry is normally like an act of cooking science. Okay, it's everywhere, it's there. It's just that whether you notice it or not. While biotechnology is the process of utilizing a biotechnological entity. So biotechnology, it stays somewhere there. Okay, there are some bits of biology and chemistry that are overlaps, but not part of biotechnology. But they're also part of chemistry that are overlaps with physics and biology. And in the middle of there is what you get for excellent science. Okay, excellent science is normally where you have all these three main features, working together to have a certain goals. A discovery, for example. Okay, but of course, I did not put math in here because math is everywhere. So you cannot exclude math from biology, chemistry, or physics. So that is where biotechnology is, and what we are learning is actually a very small subset of this overlap between biotechnology and chemistry. Okay, so the process that is involved in biotechnology and chemistry, if you were to extract out all these three main diversity that we've talked about, which is the first one is cellular technology and enzymatic technology. Okay, what we have is normally you will see a relationship between cellular technology and genetic technology. Okay, and for enzymatic technology, it normally stands on its own. So cellular technology and genetic technology is normally related to fermentation. Why fermentation? Because fermentation uses cells. And to use cells, you need a cellular technology. So you need to understand the cell. What does it do? If you change, for example, the temperature, what will it do? Will it die? Will it work faster? Or when you can actually modify the cells so that um, it only under a certain condition, under a certain external stimuli. Okay, for example, you can actually ask the cell to produce a green fluorescence protein, GFP, in the presence of glucose. Okay, so you can do multiple types of modification, and that's why it's an overlapping between cellular and genetic, because you want the cell to work in some ways, but also you can actually modify the genetics so that it works in the way that you want. Okay, and those two things, when you combine it, is what we call as fermentation. So fermentation, it's also related to a bioreactor. Okay, because bioreactor for cells is what we call as fermentation. While enzymatic technology, is an enzyme. You took it out, you use it for a certain purpose. That's it. So that's enzymatic reaction. And fermentation towards the end, it can be an in vivo expression system. It can be genetic manipulation. For genetic technology, for cellular technology, is more in vivo expression system that you can adjust. And for enzymatic, it's just either enzymatic expression from the cells, you extract it out, or you can synthesize your own enzyme certain ways that you want it. For example, a good example of an enzyme that a lot of people have been using and they are not from biological sources is insulin. Okay, so if you have a diabetes, normally you need to have an insulin injection. So this insulin nowadays normally produced synthetically or semi-synthetic, I would say. So it was not produced 100% in a cell, and it was not produced 100% from a chemical bioreactor. So that's a mix of both. Okay, so advantages of biotechnology and chemistry. First one is selectivity of the biomaterials. Okay, so because enzymes, if you use it, it can be a chemoselective, it will be a radioselective, stereoselective, and enantiomeric selective. So hopefully you can recall all of these keywords. Okay, so if you can't, then just go back and recall these words, or I know some of you guys will just simply Google it. So you can, it doesn't matter, as long as you remember what are these. So chemoselective, meaning that it's selective towards a certain environment or a certain chemical stimuli. Regioselective is a region selective. Stereoselective is you have cis and trans. And then chemoselective is RNS. Okay, so just go back and look at these keywords if you have forgotten about them. And then the other advantages is compared to a pure synthesis, it's greener, because one thing, you have less hazard or waste. Because normally when you do a biotechnology, what you need to use is you need to have an aqueous environment. Okay, so aqueous environment, of course, it's greener compared to if you use, say, for example, a pure hexane in a reaction or pure toluene, THF. So organic solvents are not green, they are harmful for the environment, harmful for aquatic environment. But if you use a biotechnological processes, you will work, you will try and produce a technology that will work in water. And if you are successful, then the byproduct is just a water byproduct or water-based byproduct. So you can just simply throw it away. Okay, and also compared to a pure synthesis, it consumes less energy. Okay, for example, a certain reaction, you need to heat it up to 100 degrees for it to work. So if you have a catalysis, if you have a catalyst that is an enzyme, So if the enzyme works at, if the enzyme comes from a human, which our body is normally around 37 degrees, so that enzyme or that reaction can now occur at 37 degrees by using enzyme. So that's what it means by it's greener in terms of producing less waste or a less hazardous waste, and the energy that is used for the same chemical reaction is less. And the third advantage is cell as a chemical factory. So when you have this to happen, if you can engineer a cell to do what you want, then it's basically you have a factory. You put in something, and then the cell produces whatever you want. So it saves a lot of time. It's like you're bringing a lot of China into a small cell, and you can do whatever you want. Okay, and what's more amazing is that a cell can do, my mouse is not working, can do a multi-step reaction. So a pure synthesis, you need to do one reaction at a time, and then take it out, clean it, and then follow it by the second reaction. But if you can produce a biotechnology, well, there are some things that can be done, but I have not done so. Okay, but the idea is there, and there are some people who have actually managed to prove that a multi-step compact synthesis can be done in a cell. Okay, some hindrances about biotechnology and chemistry. The bioreaction needs to be in thermodynamic equilibrium. The reaction medium needs to be, adjusted properly. So you cannot simply, like when you do a pure organic chemistry, what you normally do in the lab is you take ingredient A, you have ingredient B, you just mix them together, and then you just wait until the magic happens, and then you just extract the products and then purify. Okay, but for biotechnology, you do need to have a separation process. So without the separation process, normally the product will be like very difficult to, if the product is for human consumption or for human use, then the separation process is a compulsory. If it's not, then there's a bit of leeway there. Okay, and then it's always a thermodynamically equilibrium. So you need to have an equilibrium because it's an enzyme. If you're using an enzyme, if you're using a cell, you cannot heat up your reaction to say 200 degrees Celsius. It doesn't work. Or you cannot run your reaction at 0 degrees Celsius. Well, there is a chance that it will work depending on what kind of bacteria that you can use. But generally, there are limitations. Okay. Second one is biological tuning. So you have these cells or enzymes that you want to use. You always need to tune it. You always need to go back and fine tune your reaction so that it works to the exact thing that you want. Because an enzyme might work very, very easily for compound A, but then when you choose a derivative of compound A, it doesn't work anymore. So you need to fine tune it. So you need to fine tune it, either choose a different enzyme belonging to the same category, or change the temperature and whatnot. Okay, so there's a lot of process. There's a lot of things that you need to play around for the biotechnology to actually work. Okay, and the third one is isolation and separation. So again, you have your enzyme, you have your reactants, you have your products. How do you separate them and isolate the products that you want for you to use? So enzymes or cell, enzyme is in vitro, cell is in vivo. How are they compared? So these are just an example of how they are compared. You can actually refer to the textbook that is part of the references that is used for this lecture. I just take it out, extract it from there, so you can actually see some pro and cons of the enzyme and whole cell. I'm just showing it here. I can't remember whether this slide is already in your lecture notes or not. If it's not, then I will share this slide afterwards. Okay, but anyhow, this lecture is recorded, so you can just go back and have a view once I uploaded it. Okay, and finally, these are the two main references that that I'm using. So it's the main one, but it doesn't mean that all my information is limited to this. So I do use different books and different resources, websites, for example, as you will see in Wednesday. But that's about it. Okay. I think that's all for today. About 45 minutes. I'll keep that one. Um, and I think that's it. If you do have any question, you can shoot it now, or we'll just end our classes. Anyone? I'll count to 10. If you do have questions, now is the best time. Otherwise, Hidayah?
But we cannot access
Okay.
the attendance.
Okay. Try and talk again.
We cannot access the attendance.
Oh, you can access the
Yeah.
attendance. Okay, I'll have a look. Um, does it not open, or what does it say? Yeah.
Please, we need permission.
Because I tested the attendance just now, and I wasn't able to open it. So probably it's because the settings. Well, I'll change the settings soon. It's fine as long as you write your attendance roughly around this time, then it's okay. Okay. Any other questions? Okay. No. All right. So thank you, and I see you guys again on Wednesday. Bye.